everyone, how are you all? And in today's lecture, we are going to talk about an overview of cell. So let us begin with our topic. So as we know that many organisms like plants and animals of various shapes and sizes are inhabited on Earth. But they all have one thing in common, and that is they are all made up of cell, which is considered as the fundamental unit of living things. And these cells in our body performs various functions because of which we can do various activities in our day-to-day life. So scientists did not had any idea about the cell until the discovery of a microscope. So Anton von Leeuwenhoek, who was the Dutch scientist, he was the first person to discover and see the cell which is present inside an organism. So later on, Robert Brown, he expanded to the study of Anton von Leeuwenhoek and discovered the presence of nucleus inside the cell. So the pink color structure just in between the cell, it is known as nucleus, and it was discovered by Robert Brown. So in year 1838, Matthias Schleiden, who was a German botanist, he examined numerous plant species, and by examining them, he found out that plants have different cells which forms plant tissues. So now an year later, that is in year 1839, a British zoologist whose name was Theodore Schwann, he examined different animal cells and he found out that animal cells have thin outer layer which we know as the cell membrane or the plasma membrane. So here in the first picture you see the whole structure, we call it as the plasma membrane, and it was discovered by Theodore Schwann in year 1839. Now, apart from studying animal cell, he also studied the plant cells and concluded that in addition to the cell membrane, cell wall, which is a characteristic feature of plant cell, is present in the plants. So the difference he found out that in animal cells only plasma membrane is there, and in plant cells with the plasma membrane, cell wall is also present on the outermost layer of the cells. Schleiden and Schwann together formulated the cell theory, which stated that plants and animals are made up of cells and product of cells such as proteins and lipids. But the interesting fact was it did not explain the formation of new cells. So this theory was later studied by Rudolf Virchow in year 1855. So later on, Rudolf Virchow coined a Latin phrase that says omnis cellula is cellula, which means that new cells are formed from the pre-existing cells, and the same thing you can see happening in this animation on your screen. So according to this fact, he later modified the cell theory, which now states that all living organisms composed of cells and products of cells, and secondly, all cells arise from the pre-existing cells, which makes this point really important. So the cell research received a further boost after the invention of an electron microscope. And with the help of electron microscope, scientists were able to observe the structure of cell. And after observing it, they found out that the structure of cell varies in plants and animals. Now if we look at the plant cell, so we see that the plant cell is surrounded by the cell wall on the outside and the cell membrane in the inside. Whereas if we see the animal cells, so we see that animal cells lack cell wall, that they do not have cell wall present on the outside, but instead they have plasma membrane that surrounds the outer boundary of an animal cell. So now apart from the cell wall and cell membrane, both plant cell and animal cell have a spherical dense structure that is called as the nucleus. So this is the nucleus which is present in both plant cell and animal cell. So now the nucleus contains a thread-like structure which is called as chromosomes. Means this thread-like structure present or shown here on the right side, it is present inside the nucleus of plant and animal cell. Now each chromosome, all the chromosomes that you see here in this image, all these chromosomes has DNA, which is the double-stranded helical structure, looks like this. So this DNA is present in each chromosomes that you see here in this picture. The light green colored chromosome just inside this chromosome. So this is the genetic material which is inside our body, and it helps in the storage of the information and also helps to pass this information from parents to the offspring. Now in some cells, the nucleus is not enclosed in any membrane. So here in this cell, on the upper one, you see that the nucleus is not enclosed in any membrane. Not enclosed in any membrane. So the term we use for it, that here the nucleus and other cell organelles which are present are not membrane bound. Means these organelles, the various cell organelles present here, the dot-like structure you see in the cell and the nucleus, they are not enclosed in any membrane. So the kind of cells which are not enclosed in the membrane are known as the prokaryotic cell. They are called as the prokaryotic cell. And whereas the cells which have the membrane-bound nucleus and cell organelles both, as you can see in the second picture, so such kind of cells are called as the eukaryotic cells. So the examples of prokaryotic cells are the bacteria. Now bacteria do not have the nucleus which is membrane bound, and in case of eukaryotic cells, the examples are plant and animal cells. Plant and animal cells which have well developed nucleus which is also membrane bound, and all the cell organelles which is present inside the eukaryotic cells are also membrane bound. Now both prokaryotic cell and the eukaryotic cells are filled with the cytoplasm. Now cytoplasm is the semi-fluid matrix, the yellow colored present inside the prokaryotic cell and the blue colored one present inside the eukaryotic cells. Now these semi-fluid matrix are called as cytoplasm. Now these cytoplasm are present inner to the cell membrane. But in case of eukaryotic cell, the cytoplasm is not suspended inside the nucleus because it is membrane bound, but instead of cytoplasm, the fluid nucleoplasm is present inside the nucleus. The fluid nucleoplasm. So all the content of nucleus are suspended inside the nucleoplasm, but in case of prokaryotic cell, all the contents including the nucleus and other cell organelles are suspended inside the cytoplasm. So the cytoplasm present inside the cell is the hub of various activities and chemical reaction. So here the cytoplasm which is present inside the cell, we are talking about it, and the function of cytoplasm is to host various chemical reaction and activities. For example, the protein synthesis. Now this protein which is synthesized, as you can see in this animation, that the protein which is synthesized helps the cell to remain alive for a longer period of time. Now suspended in the cytoplasm are the distinct structures called as cell organelles. But as we know that eukaryotic cells are more advanced as compared to the prokaryotic cells, so in case of eukaryotic cells, you will find the well-developed membrane-bound cell organelles such as the endoplasmic reticulum, which consists of both rough endoplasmic reticulum and smooth endoplasmic reticulum, the Golgi complex or the Golgi apparatus, lysosomes, mitochondrion, the microbodies and the vacuoles. But since this is the animal cells, so you will not find the well distinct vacuoles in here. So in case of plant cells, you will find the well-developed vacuoles present in them. But in case of prokaryotic cells, the membrane-bound organelles are completely absent, means the organelles which are suspended in the cytoplasm in case of prokaryotic cells, they do not have any membrane around them. So the cell organelles, ribosomes. So ribosomes are the non-membrane bound organelles that are found in both prokaryotic and eukaryotic cells, which is found suspended inside the cytoplasm. But interestingly, it is also found within two organelles. First one is the mitochondria. Here it is the mitochondria where you can find it. And another one is the rough endoplasmic reticulum. So this membrane, this tagged membrane-like structure you see here is the structure of ER, that is also called as the endoplasmic reticulum. And on the endoplasmic reticulum, the small bead-like structure that you can find here attached to the surface of this ER are the ribosomes, which are non-membrane bound, because as you can see that these ribosomes do not have any membrane surrounded around them. So for this reason only, this ER, which is called as endoplasmic reticulum, is called as rough endoplasmic reticulum because of the presence of ribosomes on them. And in case of plant, you can find it suspended inside the cytoplasm. So the animal cells also possess a non-membrane bound organelle which is called as centrosome. So this structure, this stick-like circular structure you see here is the centrosome, and it aids in the process of the cell division. So this process cell division that we are talking about, that you are going to study later on in your subject. So now these cells might differ in their shape, sizes and activity. For example, the mycoplasma, it is the smallest cell, about 0.3 micrometer in length, and the bacteria, whereas they could be around 3 to 5 micrometer in length. Now if we talk about the ostrich egg, it is considered as the largest isolated single cell, and whereas the RBCs, they are about 7.0 micrometer in diameter. And the nerve cells, these are considered as the longest cell present inside our body. So these cells also vary greatly in shape. They may be disc-like, or maybe polygonal, cuboidal, or maybe thread-like, and even the irregular shape. So these shape of the cells vary according to the functions they have to perform. So yes, this was all about this topic. Hope you understood it well. Stay tuned for more amazing videos on EduRev. I'll see you next time. Thank you.